Hi, my name is Gyorgy Abrusan. I'm a postdoc at the University of Cambridge, and I'm going to talk about the role of allosteric proteins in genetic disease. My talk will have three parts. First, I will have a brief introduction on the concept of allostery. Next, I will show that known allosteric proteins are central in genetic disease. And finally, I will test several hypotheses on the possible causes of this phenomenon. So allostery is basically information transfer within a protein or protein complex. And a typical allosteric protein has an orthosteric site, which normally is the normal binding site of the protein, and several or one allosteric sites which act as regulators. And allosteric proteins are very important in drug design because drugs that target such sites are generally much more specific than drugs that target orthosteric sites. To analyze the importance of allostery, we compiled a dataset from several large databases like ClinVar, OMIB, UniProt, and HDMD, which altogether contains about 6,000 proteins. And we also use the disease nomenclature from the disease ontology database and the list of known human allosteric proteins from the allosteric database, which is altogether 835 proteins. Next, we perform the disease ontology enrichment analysis, which is pretty much the same as regular gene ontology analysis. Disease ontologies have a similar graph structure like gene ontologies, and what we found that allosteric proteins are enriched in a very diverse set of diseases, which include cardiovascular diseases, metabolic diseases, central nervous system disease, and a large number of cancers. On the left side, you have a plot which presents these diseases in a so-called semantic space. And altogether, we found something like almost 200 disease terms that were enriched in where allosteric proteins were enriched. On the right side, on panel B, you see a graph of the most significantly enriched diseases, which is basically dominated by hematopoietic diseases and cancers. Next, we constructed a disease protein network where every protein, where every two proteins were connected if they contribute to the same disease. The analysis of the resulting networks indicates that allosteric proteins are hubs in them, both when all proteins and when different quaternary structures are considered separately. They are involved in more diseases and have higher between centrality than other non-allosteric proteins. We also found a similar pattern is present when drug targets and non-drug target proteins are analyzed separately. Finally, we analyzed 242 cancer genome-wide association studies to test whether the loci identified by them are enriched near allosteric proteins. We found that yes, this is the case, and allosteric proteins are approximately 2 times more likely to be identified by GWAS variants than expected by chance, and this pattern is even stronger when only the most significant loci or only studies reporting fewer than 10 variants, are used. Next, we examined the possible causes of this pattern. Sequence conservation is somewhat higher in allosteric proteins, and they also have more known pathogenic mutations than non-allosteric proteins. This might indicate higher vulnerability to mutation, but also research bias. However, the relationship between the number of diseases and the number of pathogenic mutations is qualitatively similar for allosteric and non-allosteric proteins. This pattern is not different when only a single protein family like kinases or drug targets are included in the analysis. Allosteric proteins are characterized with multiple binding sites and allosteric pathways that connect them. Thus, the number of mutations that may interfere with the correct functioning of these proteins is greater than in non-allosteric proteins. We examined whether the dynamic nature of these proteins can contribute to their centrality in disease by testing whether pathogenic mutations are preferentially located in the regions that are the most important for the internal dynamics of these proteins, the interfaces of so-called communities. We found that pathogenic mutations are overrepresented in community interfaces. However, the enrichment is similar for allosteric and non-allosteric proteins. The distribution of disease mutations across communities differs more from the random expectation in allosteric proteins than in non-allosteric ones, which nevertheless indicates that pathogenic mutations do have a stronger effect in allosteric proteins. Finally, the importance of allostery in disease may be the result of the systems level characteristics of these proteins and not their biophysical properties. To examine this, we compare the centralities in disease and protein-protein interaction networks. We found that even though the overall correlation between disease and protein-protein interaction networks is low, allosteric proteins have high betweenness centralities in both, and centrality in cellular networks is likely to contribute to their importance in disease. I conclude the presentation with a summary. Non-allosteric proteins are much more important in disease than other proteins. Their dynamical characteristics probably do contribute, but are unlikely to be the main cause of this pattern. And the centrality of allosteric proteins in protein-protein interaction networks is likely to be one of the main causes. Thank you for your attention.